So, let us take for today's class the details of aerobic respiration. So, what actual aerobic respiration means? So, this is nothing but a complete oxidation of food materials like glucose into carbon dioxide and water, along with the liberation of 686 kilocalories in the case of glucose. So, total oxidation of glucose molecule and liberation of CO2, water and energy is called aerobic respiration. So, basically it is an exergonic process. Why it is called exergonic process? Since energy is liberated. So, as energy is liberated, one can call this metabolic process as an exergonic process. And during this respiration, what happens? Glucose is broken down into simple. That means food is going to be destructed. So, one can call it is a catabolic process. So, you know that metabolism in living cells completes either in anabolic way or catabolic way. So, catabolism is a kind of a destructive process, whereas anabolism is a kind of constructive process. So, photosynthesis is a constructive process, whereas a respiration is a destructive process. So, since here food molecules are destructed, so breaking down of food. So, it is called catabolic, and during respiration chemically it is an oxidation process where hydrogens are removed during respiration. So, chemically respiration is an oxidation, and basically respiration is a catabolic and exergonic process. Because of this respiration, the dry weight of organism decreases. So, that implies this is a catabolism. So, reason for decreasing the organism's dry weight during respiration is catabolic process. As respiration is a catabolic process, the energy food molecules are destructed. So, dry weight of organism decreases. And coming to the details of aerobic respiration, in aerobic respiration there is a complete oxidation of glucose, which is a simple respiratory substrate. Otherwise, the most common type of respiratory substrate. And this glucose undergoes oxidation at 6 steps. So, there are 6 oxidations occur during aerobic respiration. That means one molecule of glucose undergoes 6 oxidations, and there is a liberation of 6 CO2 molecules, and during this respiration there is a formation of 12 H2O molecules and release of 686 kilocalories of energy, and where 6 oxygen molecules are utilized. So, let us take the summary. During aerobic respiration, one molecule of glucose is consumed, 6 molecules of O2 utilized, 6 CO2 molecules liberated, and 12 water molecules are released, and 686 kilocalories of energy is released. So, this is the summary. So, let us take the what equations occur in aerobic respiration. Let us take the various steps that occur during aerobic respiration, and during those steps what events occur and where those events occur. So, a glance of aerobic respiration. There is the first step in aerobic respiration is known as glycolysis. So, glycolysis is the first step in aerobic respiration. This is the process where the glucose molecule converts into two molecules of pyruvic acid. So, simply glycolysis, the first step of aerobic respiration, involves conversion of glucose, a C6 compound, into two molecules of C3 compounds called pyruvic acids. Where it occurs? Already respiration is a process usually occur in cell, either eukaryotic cell or a prokaryotic cell. But first step of aerobic respiration always occurs in cytosol. So, otherwise cytoplasmic matrix. So, the fluid matrix of cytoplasm, otherwise called cytosol. So, one place. And another step in aerobic respiration, of course, called second step, which is called more popularly as a link reaction. A link reaction is a second step in aerobic respiration, which involves oxidative decarboxylation of pyruvic acid. Oxidative decarboxylation of pyruvic acid into acetyl coenzyme A. So, let it be like this. The pyruvic acid undergoes oxidation, the removal of hydrogens, undergoes decarboxylation, liberation of CO2, and condenses with coenzyme A and forms acetyl coenzyme A. So, this is considered to be the second step in aerobic respiration, where pyruvic acids, the end product of glycolysis, undergoes some complex conversions, loses hydrogens, loses carbon dioxide, and reacts with coenzyme A and forms acetyl coenzyme A. So, it occurs in mitochondrial matrix. So, after the first event, the next process of aerobic respiration shifted from cytosol to mitochondrial matrix. So, mitochondria is a cell organelle, of course, important organelle for respiration. This is a well known organelle. You might have studied the details of mitochondria in cell biology. So, matrix of mitochondria is a site for link reaction. So, another step in aerobic respiration is called Krebs cycle, very popularly called as TCA cycle. So, what happens during Krebs cycle? So, during the Krebs cycle, the end product of link reaction called acetyl coenzyme A undergoes complete oxidation and decarboxylation into CO2 and water plus some reduced coenzymes like NADH pluses and FADH2s. So, complete oxidation and decarboxylation of acetyl coenzyme A takes place during Krebs cycle, and this is a process takes place again in mitochondrial matrix. So, two events in aerobic respiration occurs in mitochondrial matrix. So, acetyl coenzyme A converts into CO2 and water along with the formation of some reduced coenzymes like NAD, H plus and FADH2. And the last step in aerobic respiration, of course, the fourth and final is very important, which is called as ETS, coupled with oxidative phosphorylation. So, what is the fourth step? ETS, that is electron transport system, along with oxidative phosphorylation. This is the step where the reduced coenzymes, what are they? NADH and FADH2s are oxidized. So, reduced coenzymes which are formed in various steps undergoes oxidation and also reduction of oxygen and followed by formation of ATP or synthesis of ATP. So, last and final step where the coenzymes are oxidized, oxygen is reduced and ATP is synthesized, and this is the process that takes place on inner mitochondrial membrane. So, you know that mitochondria made up of two membrane envelope, outer and inner. So, inner mitochondrial membrane is the site where the last and final step of aerobic respiration occurs, and it may be folded and those are called cristae. So, let it be more correct. Cristae are the sites of last and final step of aerobic respiration. So, 4 different steps. In all these 4 steps, if you go details, only one step where the oxygen is involved. So, basically what aerobic respiration? The complete oxidation of glucose in the presence of oxygen, but oxygen role during this process seen or occurs only in the last step, but not either first or second or third. So, that is another interesting feature of aerobic respiration. It is a process where oxygen is required, but not in all steps. The last event is the process where direct utilization of oxygen, even though this process is called aerobic respiration. There are already discussed regarding the aerobic. So, there are 6 oxidation steps altogether in aerobic respiration. Let us see where these oxidation steps occur. So, during glycolysis, there will be only one oxidation. In link reaction, one more oxidation. So, remaining 4 oxidation steps completes during Krebs cycle. So, out of 6 oxidation steps that usually occur in aerobic respiration, 1 oxidation in glycolysis, 1 more oxidation in link reaction, and 4 oxidations occur in Krebs cycle. So, out of these, first oxidation completes in cytosol and the last 4 completes in mitochondria. So, more number of oxidations occur during mitochondria. That is why mitochondria considers the important cell organelle for respiration. Okay, student. Now, we will discuss about the first and important step of aerobic respiration called glycolysis. So, glycolysis is an event in respiration, because if you take the origin of the term glycolysis, in Greek language, glykos means the sugar, whereas lysis means destruction or breakdown. So, by combining these two words, glycolysis is a process where a sugar is destroyed or broken down. So, destruction or breaking of sugar takes place during the first step called glycolysis. So, let us take the importance of glycolysis. Glycolysis is a step that occurs both in aerobic respiration and anaerobic respiration. So, that means this is a common step in both aerobic and anaerobic, and it is also commonly called as core respiration. Since it is occur commonly in both types of respiration, some scientists name it as a core respiration. And during glycolysis, the hexose molecule, say glucose, is converted into two molecules of pyruvic acid. And during this process, there is utilization of ATP, there is liberation of removal of hydrogens, which are utilized in the reduction of NAD plus into NADH plus H plus, and there is a synthesis of ATP. So, if you see the summary of glycolysis, an hexose molecule converted into two molecules of C3 compound pyruvic acids, during which ATP is utilized, ATP synthesized, and hydrogen atoms are removed to form NADH plus H plus. So, by seeing this, one can say the glycolysis is not going to be complete in one step. So, there are several steps which occurs in a sequential manner during glycolysis. Altogether, there are 10 reactions which involves in a serial order the conversion of a big hexose molecule into two simple organic molecules. So, there are 10 reactions, and all these 10 reactions that are involved during glycolysis were discovered by 3 German scientists by name Emden, Meyerhoff, and Paranas. Emden, Meyerhoff, and Paranas. So, to honor the discoverers, this process is renamed as EMP pathway. So, E stands for Emden, M stands for Meyerhoff, and P stands for Paranas. So, glycolysis, another name is EMP pathway. Most of the cellular organisms, this glycolysis takes place by a mechanism called EMP pathway. So, this is the introduction of glycolysis. So, see what are the details of glycolysis. So, generally in glycolysis the common substrate is glucose, but glucose is formed from different sources, sometimes starch or sometimes sucrose, since in plants the main reserve food is starch, one form of polysaccharide, and another one is another form of polysaccharide available in plants is sucrose. But starch and sucrose are unfit for the respiratory process. They cannot directly oxidize since they are big molecules. So, for the initiation or initial step of glycolysis or respiration, glucose is to be present. So, starch converts into glucose or sucrose may converts into glucose. So, after the conversion of either starch or glucose in or sucrose into glucose, there starts the aerobic respiration. Generally, amylases involves in the digestion of starch, whereas invertases involves in the conversion of a disaccharide like glucose into disaccharide sucrose into glucose or fructose. So, before the actual oxidation of glucose, there is a digestion. Starch converts into glucose or sucrose broken down into glucose and fructose. So, invertase is an enzyme which act upon sucrose, which is a disaccharide, and it converts into two different types of hexoses. So, after that the respiration starts. So, glucose is an initial substrate for respiration. It comes either from starch or sucrose after some enzymatic reactions. So, let us take that the actual process of glycolysis. So, in this case glucose converts into glucose 6-phosphate. So, glucose is a C6 compound which is converted into glucose 6-phosphate. That means a phosphate is added to glucose at a 6th position and called glucose 6-phosphate, for which phosphate must be donated, and ATP already present in the cells donates the phosphate and converts into ADP. So, this is a process of phosphorylation where ATP transfers one of its phosphate to an exose simple molecule called glucose, which converts into glucose 6-phosphate. And this is a process where two substances are formed, glucose to glucose 6-phosphate, in the mediation of an enzyme called hexokinase. As already stated, that metabolic process are mediated by enzymes. So, the first reaction in glycolysis reads like this: glucose plus ATP gives rise glucose 6-phosphate plus ADP with the presence of an enzyme hexokinase. Hexokinase is a kind of metalloenzyme. That means it is a complex or a compound enzyme which requires a metal ion as a cofactor. So, there are some types of enzymes, holo enzymes and simple enzymes. Already you might have seen that in enzymes. So, hexokinase is a holo enzyme which contains a coenzyme part and a cofactor, and the cofactor in this enzyme is a metal ion, magnesium. So, in the presence of magnesium, hexokinase converts the simple hexose molecule into a phosphorylated hexose molecule, which is also C6. So, this glucose 6 phosphate is converted into fructose 6 phosphate, so which is also C6. Glucose and fructose, as you know, they are isomers. So, glucose isomerized into fructose. So, there is no question of addition of any group or deletion of any group, simply intramolecular group transfers. That reaction is catalyzed by hexose phospho isomerase. So, hexose phospho isomerase is an enzyme. In the presence of this enzyme, there occurs a second reaction in glycolysis, which reads glucose 6 phosphate converts into fructose 6 phosphate in the mediation of hexose phospho isomerase. So, this fructose 1,6 phosphate is converted into fructose 1,6 bisphosphate. So, as another intermediate is formed with 6 carbons and 2 phosphates. One phosphate is at first carbon of this skeleton and another phosphate is the 6th carbon of the skeleton. As in a compound, if 2 carbons are placed at 2 different places, as per the nomenclature, it should be called bis. Instead of di, it is bis. So, fructose with 6 carbons, where first carbon and 6th carbon has phosphate. So, where from that extra phosphate comes? Once again, that phosphate is donated by one more ATP molecule and it converts into ADP. So, there is another step where ATP is utilized. So, this is the process catalyzed by an enzyme phosphofructokinase. Phosphofructokinase, and this is also an holoenzyme which requires magnesium help. So, hexokinase is an enzyme with magnesium cofactor. Phosphofructokinase is another holoenzyme with a magnesium as a cofactor. Now, a hexose molecule with 2 phosphates are formed. So, this is a highly energetic molecule ready for breaking. So, first reaction and third reaction in EMP pathway are very similar in one aspect. What is it? Formation of ATP, utilization of ATP, removal of hydrogens or addition of hydrogens. So, the answer is utilization of ATP. So, this is an interesting point. As already stated that respiration is a process for generation of ATP, but so far we have seen ATP are utilized instead of production. So, this is going to be a business. Any business supposed to be start with an investment. So, in order to get profits in a business, whatever it may be, one must invest. So, cell also invest 2 ATP molecule for extraction of energy locked in glucose. So, ATP utilized in the first 2 steps, that is first step and third step. And so, scientists named this reactions as preparatory phase. preparatory phase. So, this C6 compound, that is with two phosphates, finally broken down into two C3 compounds. One is called phosphoglyceraldehyde, let it be PGAL, phosphoglyceraldehyde, sometimes may be called 3-PGAL, 3-phosphoglyceraldehyde, or G3P, glyceraldehyde 3-phosphate. There are different names, 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate, which is a C3 compound. And another one is dihydroxyacetone phosphate, abbreviated as DHAP, dihydroxyacetone phosphate, and G3P are the compounds formed from a big C6 molecule during glycolysis. So, it is physically breaking up a C6 compound into C3 compound, and it is catalyzed by an enzyme aldolase. So, the fourth step, so far how many steps? 4 steps. The fourth reaction in EMP pathway is catalyzed by an enzyme, name it aldolase. So, the fourth reaction of glycolysis results of products. What are they? So, one is G3P, another one is DHAP, and these two molecules are isomers. So, PGAL and DHAP are isomeric compounds. But coming to the actual event, among the two groups of glucose, only PGAL is a suitable compound for actual oxidation. So, what I mean, PGAL is the right kind of glucose form ready to undergo oxidation, but not DHAP. That is why DHAP molecule may convert into PGAL by means of an isomeric reaction. So, that is catalyzed by a triose phosphate isomerase. So, another reaction in glycolysis is conversion of DHAP into G3P in the presence of glyceraldehyde ketone isomerase or simply triose phosphate isomerase. So, from here onwards, the G3P available in how many molecules? 2. So, 1 fructose, 1,6-bisphosphate cleavage into a PGAL and DHAP, right? And DHAP is not suitable for further conversions. Hence, the DHAP being isomer of PGAL is converted into G3P and available for further reactions. So, let us take the further conversions. So, G3P is now suitable for oxidation. So, these G3P molecules are now available in 2. So, let us take 2 every time from here onwards. So, 2-PGAL reacts with an inorganic phosphate, which is available in H3PO4, and loses hydrogen atoms and converts into 1,3-bisphosphoglyceric acid. So, PGAL plus H3PO4 plus NAD plus, because the hydrogens removed at this stage accepted by NAD plus, which is converted into NADH plus H plus and forms 1,3-bisphosphoglyceric acid, which is another C3 compound. So, another intermediate in glycolysis is a C3 compound 1,3-bis-PGA, which is formed from G3P after a phosphorylation and an important reaction, oxidation. So, for the first time in glycolysis, there occur an oxidation. As you know, oxidation involves removal of hydrogen. So, it can be also called dehydrogenation, not dehydration. So, there is a difference between dehydration and dehydrogenation. Removal of water is dehydration, but removal of hydrogen atom from a substrate in order to form another substrate is called dehydrogenation. So, first biological oxidation step in aerobic respiration occurs when dash converts into dash. So, that is when G3P is converted into 1,3-bis PGA, there is an oxidation, and these hydrogen atoms taken by NAD plus nicotinamide adenine dinucleotide, which is an important coenzyme. It is an important coenzyme, acts as an hydrogen acceptor and reduced into NAD H plus H plus. So, there are two PGAL molecules. So, there are two NAD pluses converts into two molecules of NAD H plus. So, NAD H plus is a one form of energy. Unfortunately, it is not useful. So, glucose energy in the form of electrons and proton locked in two molecules of NADH. Further energy is still in the 1,3-bis PGA, but later this NADH is finally oxidized and involves in ATP synthesis comes later. But this is the enzyme which control this reaction is also called G3P, glyceraldehyde 3-phosphate dehydrogenase. So, glyceraldehyde 3-phosphate dehydrogenase catalyzes the reaction PGAL into 1,3-bis PGA. So, where phosphate is added, but interestingly that phosphate is not from ATP, but from inorganic. Next, this 1,3-bisphosphoglyceric acid loses phosphate and which is reacted with ADP converts into ATP. See, this is the first time in respiration ATP is generating when 1,3-bis PGA is converting into PGA. So, chemically this is the reaction called dephosphorylation, where phosphate is removed, added to ADP, converts into cell currency ATP. So, another C3 molecule is formed which is called 3-phosphoglyceric acid. See, first phosphate is removed, taken by ADP, converts into ATP. The rest of the molecule can be termed as 3-phosphoglyceric acid. How many molecules are here? Two molecules of 1,3-bis PGA after losing phosphate converts into two molecules of ATP, and this is the reaction catalyzed by phosphoglycerokinase. Phosphoglycerokinase is an enzyme which catalyzes this reaction, conversion of 1,3-bis PGA into 3-PGA. Again, magnesium is the cofactor. So, this formation of ATP when one substrate converts into another substrate without participation of ETS is well popularly known as SLP. What SLP stands? Substrate level phosphorylation. So, is the one type of ATP formation occurs in cells without electron transport involvement, system involvement. If ATP is generated at substrate level, it is called SLP. So, in glycolysis, the first SLP step mediated by, so answer is phosphoglycerokinase. And 3-PGA now converts into another intermediate called 2-PGA, where only group transfer. The third carbon of PGA is now with phosphate, which is shifted to second carbon. So, intramolecular group transfer, otherwise called the frame shift, otherwise called the group transfer, which is catalyzed by an enzyme phosphoglyceromutase, where 3-PGA is converted into 2-PGA, is one kind of isomerization. And now 2-PGA is going to be lose water and converts into phosphoenol pyruvic acid. So, there is a dehydration, not dehydrogenation, dehydration in the presence of an enzyme enolase. So, enolase is an enzyme which catalyzes dehydration reaction, involves conversion of 2-PGA into PEP, which is another C3 compound. Finally, two water molecules. So, 2 molecules of phosphoenol pyruvic acid undergoes one more time dephosphorylation, taken by 2 ADP, converts into 2 ATP. So, one more time there is an SLP step, where one more time ATP is formed. Finally, a compound with 3 carbons is called pyruvic acid. So, finally, one molecule of glucose through this mechanism converted into 2 molecules of PEP, 2 molecules of pyruvic acid. So, during this process, 2 ATP molecules were utilized, 4 ATP molecules are formed, and 2 NADHs are also generated, and 2 water molecules are released. So, that is the summary or the details of glycolysis. So, let us take the summary of glycolysis. So, what are the points in glycolysis? So, during glycolysis, glucose is synthesized or consumed. Definitely it is utilized. Glucose is utilized. And during glycolysis, what about ATP? So, ATP molecules during glycolysis are utilized and also formed. So, there is a formation of ATP and utilization of ATP both occurs during glycolysis. And during glycolysis, what about oxygen? What about carbon dioxide? So, see, after seeing all 10 reactions, step 1 to last reaction, there is no reaction where O2 is utilized, and also there is no reaction where CO2 is not liberated. So, O2 is not utilized, CO2 is not liberated. Though you are discussing the aerobic respiration, oxygen is important, but interestingly O2 is not utilized during glycolysis. So, one can say glycolysis process requires or it involves an anaerobic process. So, without oxygen the glycolysis process is able to complete, and during glycolysis the end products are what? The end products of glycolysis, 2 molecules of a C3 compound, that is pyruvic acid, and 2 molecules of NADH plus H plus, one form of energy to be oxidized later to get ATP, and 2 molecules of water is formed, and 2 molecules of ATP utilized, 4 molecules of ATP formed. So, the end products of EMP pathway, formation of 2 pyruvic acids, results of 2 NADH plus H pluses, 2 water molecules, 2 ATP are utilized and 4 ATP are formed. That means the net gain of ATP is 4 minus 2. So, during glycolysis there are only 2 ATP released as a net. So, 4 are formed, 2 are utilized. So, finally, net gain is 2 ATP. And during the glycolysis of aerobic respiration and the glycolysis of anaerobic respiration, as I already discussed that glycolysis is a common step in both. So, in aerobic respiration, for 1 molecule of glucose, 2 ATP consumed, here also 2 ATP consumed. 4 ATP are formed as through SLP. Here also 4 ATP are formed through SLP. And in aerobic respiration, later the 2 NADH plus H plus through ETS yields 4 more ATP. So, through oxidative phosphorylation, the NADHs formed in aerobic glycolysis involves in oxidation steps through ETS and forms 4 more ATP. But in anaerobic respiration, NADH pluses are not participate in ETS. So, there is no extra ATP formation. So, finally, in aerobic glycolysis, considering the SLP and oxidative phosphorylation. So, total ATP formed in aerobic glycolysis, take 4 ATP through SLP and 4 ATP through oxidative phosphorylation, that is 8 ATP. But used ATP 2. So, finally, the net ATP in aerobic glycolysis is 6. So, 2 types of questions. In aerobic glycolysis, the total ATP 8, net ATP 2. But in anaerobic respiration, the total ATP generated 4 and utilized 2, net ATP is finally 2. So, there is a less amount of ATP generation during anaerobic glycolysis, more ATP relatively generated during aerobic glycolysis. So, the ATP formed through SLP plus ATP formed through oxidative phosphorylation together yields 8 ATP. As usual, 2 ATP are utilized in the first and third step. So, net ATP in aerobic glycolysis. Remember, aerobic glycolysis net yield is 6, anaerobic glycolysis net yield is 2. Is it clear? So, let us take the overall equation of glycolysis. So, in glycolysis, the main participant is glucose, the C6, where 2 ATP are utilized, 2 H3PO4 are also consumed, 4 ADP are taking part at different reaction, and 2 NAD plus, which is what is it? Hydrogen acceptor, yields 2 molecules of pyruvic acid, some energy of glucose still locked in pyruvic acid, and 2 ADP, 4 ATP are formed, and 2 water molecules are liberated. So, 2 ATP are utilized, 4 ATP are formed along with 2 molecules of NAD H plus H plus. So, utilized number 2, formed number 4. So, only in glycolysis 2 ATP is net. If oxygen is available, these NADHs formed in glycolysis take part in oxidative phosphorylation through ETS, then they also yield ATP. So, let us take the detailed list of reactions that occur during this EMP pathway. So, you try to fill these blanks and read carefully. First reaction, a C6 compound glucose is the substrate plus ATP in the presence of an enzyme. What is it? Hexokinase with magnesium forms products like what? G3P is 6 compound and ADP. Chemically, the first reaction is called phosphorylation, since phosphate is added to the main substrate. Is it clear? Next, coming to the second one. So, second reaction occurs after the first. Naturally, in a metabolic process, the end product of previous reaction going to be the substrate for second reaction. So, take that glucose 6 phosphate, which is a C6 compound, in the presence of an enzyme phosphohexose isomerase converts into another hexose fructose 6 phosphate. So, enzyme is an isomerase, so reaction is isomerization. So, next second reaction in glycolysis is chemically isomerization. The substrate for second reaction hexose, the product of second reaction will also be hexose, but glucose and fructose. Next, third one, a third reaction where fructose 6 phosphate going to react with something. What is it? Another ATP. So, in the presence of a third enzyme in sequence, phosphofructokinase, the ATP is transferred to fructose. So, forms fructose 1,6 phosphate and ADP. So, products of third reaction in glycolysis, fructose 1,6 phosphate and ADP. This is chemically phosphorylation. So, third step is phosphorylation. And fourth step, fructose 1,6 bisphosphate, a C6 compound, in the presence of an enzyme called aldolase, splits, not condenses, splits into 2 C3 molecule, one is G3P, so automatically other one is DHAP. So, splitting more literally cleavage, and then this DHAP is going to be converted into G3P in the presence of G3P dehydrogenase, G3P isomerase, otherwise triose phosphate isomerase. So, another reaction chemically isomerization. Since DHAP is unfit for oxidation, G3P is more fit for oxidation. So, DHF should convert into G3P. So, altogether with the completion of fifth reaction, how many G3Ps are available? 2. So, let us take 2 molecules of G3P, 2 H2PO4 and 2 NAD plus in the presence of an enzyme G3P dehydrogenase. There is a removal of hydrogens from G3P, acceptance of hydrogen by NAD plus. So, forms NAD H plus H plus, 2 molecules of 1,3-bis PGA and 2 molecules of NADH. So, this is the most important reaction in this entire EMP, oxidation. So, first oxidation step in aerobic respiration. Then later, the 1,3-bisphosphoglyceric acid, 2 molecules reacts with 2 ADP in the presence of phosphoglycerokinase, converts into 2 molecules of PGA plus 2 molecules of ATP. So, ATP is formed for the first time in the 7th reaction, and it is a dephosphorylation reaction, otherwise SLP. So, the 7th step in sequence in glycolysis is SLP, substrate level phosphorylation. And 8th one, 2 molecules of 2-PGA in the presence of phosphoglycero acid mutase. Phosphoglycero acid mutase converts into 2-PGA, which is another reaction, otherwise called frame shift reaction or intramolecular group transfer. So, 2 molecules of PGA now converts into 2 molecules of PEP with 2 water molecules released in the presence of enolase. So, it is chemically dehydration. So, last but one step in glycolysis is a chemically dehydration. So, finally, 2 molecules of PEP in the presence of 2 molecules of ADP with enzyme mediation pyruvic kinase converts into 2 molecules of pyruvic acid and 2 molecules of ATP. So, substrate for last and final step of glycolysis, PEP and ADP, products of last reaction of glycolysis, pyruvic acid and ATP. So, it is another process called phosphorylation or dephosphorylation. So, SLP. So, 7th step and 10th steps are SLP reactions. So, final products are 2 molecules of pyruvic acid, 2 plus 2, 4 ATP molecules, 2 plus this one 2 and 2 molecules, 4 ATP, 2 NADH plus. So, this is a concise table useful for number of bits, substrates, products, enzymes, and correct sequence of enzymes involved in glycolysis is one type of question, and a chemical nature of first reaction, chemical nature of penultimate reaction or ultimate reaction.